In this video, we'll discuss about cultivation of viruses or isolation of viruses. Viruses is obligate intracellular organism. It cannot grow artificial culture media. It needs animal inoculation or egg inoculation or tissue culture. Cultivation of virus not routinely used in diagnostic virology, not widely used method in diagnostic virology because technically demanding, labor intensive and time consuming. It is not actually done, but exam point, this one is very important topic. Also collection of specimen, then properly transport immediately, then refrigeration is essential. Refrigeration is essential. Virus are heat liable. So considering these things, isolation or cultivation of virus not routinely used in diagnostic virology. It is used for research and vaccine trials, etc. So first talk about first option, animal inoculation. This animal inoculation because of ethical issues, animal inoculation is restricted. However, for research purpose, these still used for animal inoculation. Like if we want to study pathogenesis of virus, or they want to study oncogenesis of virus, or they want to study vaccine trials, then used animal inoculation. For animal inoculation, they actually used for infant suckling mice. So for animal inoculation, there are using infant suckling mice. This one intracerebrally inoculated or intraperitoneally they are inoculated. Two way of inoculation, intraperitoneally or intracerebrally they inoculated, and these mice are observed for signs of the disease. They observed carefully for what changes happen in the mice after that inoculation. Any changes they want to note it that way. That's why they observed in carefully. Then tissue section also taken and observed histological examination. Then basically some viruses are very hard to cultivate. Example, arbovirus and Kawasaki virus. They are very hard to cultivate. So they used for animal inoculations. So this one they are using for research purpose. Some viruses cultivate very hard. Example, arbovirus including chikungunya virus, Japanese encephalitis virus, dengue fever, then Casein or forest disease virus and rabies. These are the example of arbovirus. Kawasaki virus, it is a picoRNA virus. So second method, egg inoculation method. In this method, embryonated chick egg or duck egg there are used. So chick egg or duck egg can be used. Basically what happened here, specimen are inoculated into 4 different route. Specimens are inoculated into 4 different route. Here look at that route here. This one is embryo. This one is the embryo. This one is yolk sac. Around embryo, amniotic amniotic cavity is present. Around this amniotic cavity, around this one, allentonic cavity is present. Then all around this we have chorioallantoinic membrane. All around this chorioallantoinic membrane is presented. After that, some air shock shell membrane, then albumin also presented here. So this is a embryonated hen egg. Basically, there are 4 route inoculating of this embryonated egg. First one is yolk shock. In this yolk shock, examination is done for Japanese encephalitis viruses, some bacteria like rickets and chlamydia. So bacterial inoculation also will be done here. Then next, amniotic here, amniotic cavity is used for primarily isolation of influenza virus. Basically, viral growth of influenza can be detected by hemagglutinin. Mainly influenza virus, all 3 genome A, B, C, all the genome can be cultivated in amniotic cavity. Then allentonic cavity, only influenza virus A type only. So now coming to third one is allentonic cavity. Here, what are the things we can cultivate? This this one is the law, and comparing to other, this one is the bigger one, and comparing to other, this one is the biggest cavity. Actually, we are getting better virus yield, the vaccine yield, better yielding of viral vaccination, which are egg derived vaccine. So influenza virus A, then rabies vaccine, yellow fever vaccine make from here. Duck egg, larger one, it give better yielding. Then chorioallantoinic membrane, this one chorioallantoinic membrane. Here it is just a membrane. We are going to inoculated, or here what virus? Here box virus. So here box virus, variola vaccinia virus, then variola virus, vaccinia virus, then herpes simplex virus. Basically, this will produce the visible lesion. So after that, here it will producing visible lesions, such as box on chorioallantoinic membrane. So here we can see that box lesions. Now coming to tissue culture. Tissue culture, first one is we want to discuss organ culture. Look at this organ. This one is a tracheal ring. This one trachea ring. This one previously used of sudden fastidious virus. Previously used method, they have specific affinity for specific organ. Specific affinity for specific organ. So some virus have some affinity with specific organ. Example, coronavirus. Coronavirus have some affinity for tracheal ring. So trachea ring culture for isolation of coronavirus, that was about that organ. Previously used method. Next, explant method and cell lines we will discuss in next video. Thanks for watching.